Imagine each cell as a tiny metronome ticking away with its own biochemical beat, driven by feedback loops of gene expression, protein modification, or metabolic cycling. But in tissues and microbial communities, those cellular clocks often march in lockstep. How do they coordinate their internal rhythms to produce a unified chorus? Have you ever noticed how fireflies in a meadow begin flashing in unison as dusk falls? That same magic of coupled oscillators happens inside our bodies. In the brain's suprachiasmatic nucleus, SCN, thousands of neurons each carry a tilde 24-hour cycle. They share signals, tiny bursts of neurotransmitters or electrical impulses that gently nudge each neighbor into phase. If one neuron fires just a bit early, its neighbors get a tickle that shifts their own clock slightly forward or backward, and over time, all 20,000 SCN cells synchronize to sunrise and sunset. But what about single-celled organisms? In yeast, glycolysis itself can oscillate, with levels of certain metabolites rising and falling every few minutes. Question: can a droplet of yeast cells really pulse together? Yes. Through quorum sensing, each yeast cell leaks out metabolites into the medium. When the concentration of, say, acetaldehyde crosses a threshold, it triggers its neighbors to adjust their metabolic gears. The result is a communal heartbeat of chemical concentrations, visible as synchronized flashes of NADH glow under a microscope. In developing embryos, the segmentation clock choreographs how vertebrate bodies lay down repeated segments. Presemitic mesoderm cells each carry a fast-tilled 90-minute oscillator of gene activity. They talk to each other via the notch signaling pathway. When one cell's notch ligand spikes, it nudges its neighbor's clock hands a bit. Step by step, wave upon wave of synchronized gene pulses sweep through the tissue, marking out the rhythmic pattern of future vertebrae. Could we build our own biochemical orchestra? Synthetic biologists have crafted the repressilator, a synthetic genetic circuit in bacteria that cycles through 3 repressor proteins. By placing many of these engineered cells in a droplet and allowing them to exchange a small signaling molecule like HL, researchers see the whole population blinking in synchrony, proof that simple coupling rules can unite man-made oscillators too. So what's the secret sauce? At its heart, synchrony emerges from feedback plus communication. Each cell's internal feedback loop sets its natural pace, while even a whisper of a neighbor's signal tweaks that pace just enough. Over hundreds or thousands of whisper exchanges, the group locks into a single rhythm. By tuning the strength and timing of those couplings, biology ensures that hearts beat together, insulin pulses match blood sugar demands, and microbial mats dance in time. An elegant symphony of life's internal clocks. Biochemical oscillators are networks of chemical reactions in cells that produce rhythmic, periodic changes in concentration or activity of key molecules. These cycles can range from milliseconds to, e.g., calcium spikes, to hours, e.g., circadian rhythms, and regulate processes like metabolism, cell division, and gene expression. They arise from feedback loops, typically involving delayed negative feedback and nonlinear kinetics. Oscillators act as internal timers, enabling cells to anticipate environmental changes or maintain internal order. Their robustness and tunability make them fundamental to cellular homeostasis. The circadian oscillator in mammalian cells is governed by transcriptional feedback loops involving clock, BMAL1, PR, and CRY proteins. It's like a set of molecular alarm clocks ringing at different times to orchestrate daily cellular activity. In multicellular organisms, or even within a single cell, multiple oscillators need to synchronize to ensure coherent behavior. Desynchronization can lead to dysfunction such as metabolic imbalance, arrhythmias, or circadian misalignment. Synchrony allows tissues to perform collective tasks like hormone release, immune response, timing, or tissue regeneration. Even in unicellular populations such as yeast or bacterial biofilms, synchronized oscillators enable quorum sensing and metabolic cooperation. Maintaining synchrony is essential for coordinating time-sensitive biological processes. In pancreatic islets, Synchronized calcium oscillations among beta cells regulate pulsatile insulin secretion. It's like musicians in an orchestra keeping time, not by watching a clock, but by listening to each other. Within single cells, multiple biochemical oscillators, such as calcium waves, metabolic cycles, and mitotic clocks, can couple via shared components or feedback. Crosstalk between pathways allows phase locking, where oscillators with different frequencies synchronize or entrain one another. Shared regulators, such as kinases or ion channels, create biochemical hubs that coordinate timing. Some systems exhibit hierarchical control, where slower oscillators modulate faster ones. This internal dialogue keeps cellular rhythms aligned and avoids chaotic interactions. Mitochondrial metabolic oscillations and cytosolic calcium pulses entrain each other via NADH-dependent enzymes. It's like gears in a clock, different sizes, different speeds, but all turning in harmony through shared axles. In multicellular systems, biochemical oscillators across neighboring cells synchronize via signaling molecules, gap junctions, or mechanical cues. This coordination enables tissues to function as rhythmic units critical in heartbeats, brainwaves, and hormonal pulses. Gap junctions allow direct ion and metabolite flow between cells, promoting instantaneous phase alignment. Alternatively, diffusible signals like ATP, NO, or cytokines can entrain distant oscillators through Parkran feedback. Mechanical coupling via cytoskeletons or extracellular matrix tension can also contribute to rhythmic alignment. In the superchiasmatic nucleus, SCN, 20,000 neurons synchronize circadian rhythms through neurotransmitters like VIP and GABA. It's like dancers syncing steps by watching, listening, and feeling the stage vibrate. Biochemical oscillators can behave like coupled nonlinear systems, showing behaviors such as phase locking, synchronization, resonance, or chaos. When two oscillators interact, one may entrain the other, forcing its period, or both may shift toward a common rhythm. The strength of coupling, delay in feedback, and frequency mismatch determine synchronization outcomes. Mathematical models like the Kuramoto model are used to study collective oscillator behavior. This approach has explained synchrony in heart tissue, circadian networks, and glycolytic cycles. Yeast cells undergoing glycolysis can entrain to a common NADH oscillation frequency via acetaldehyde signaling. It's like a room full of pendulums slowly swinging in sync after enough time on a shared table. Biological systems are inherently noisy. Molecular fluctuations and external perturbations challenge oscillator stability. Despite this, many cellular oscillators are robust to noise and even use it to enhance adaptability. Weak noise can induce coherence, stochastic resonance, help prevent synchronization failure, or allow flexible phase resetting. Redundant feedback loops and amplitude thresholds buffer against random fluctuations. Cells also exploit noise to diversify responses while maintaining overall rhythm. Circadian rhythms in single mammalian cells remain stable despite fluctuations in gene expression and protein turnover. It's like jazz musicians improvising within a beat, variation within structure, not chaos. Advanced tools allow researchers to monitor and manipulate oscillations in real time. Fluorescent reporters track oscillatory proteins, metabolites, or ions at single-cell resolution. Optogenetics and chemogenetics can externally entrain or perturb oscillators with high precision. Microfluidic arrays and organoids model synchronized tissue behavior under controlled conditions. Mathematical modeling bridges experimental data with predictive dynamical systems theory. Light pulses are used to reset circadian rhythms in SCN neurons expressing light-sensitive ion channels. It's like tuning biological metronomes with a laser pointer. One pulse, many resets. Disrupted oscillator synchrony is implicated in diseases such as insomnia, metabolic syndrome, epilepsy, and cardiac arrhythmia. In cancer, cell cycle oscillators may desynchronize, contributing to uncontrolled proliferation. Neurological disorders like Parkinson's and Alzheimer's involve breakdown of central clock coordination. Therapeutics targeting synchronization, chronotherapy, electrical entrainment, or optogenetic modulation are emerging strategies. Understanding how cells coordinate rhythms offers insight into developmental timing, tissue regeneration, and aging. Chronotherapy aligns cancer drug administration with tumor cell cycle peaks to increase efficacy. It's like delivering medicine when the body is most ready to receive. Timing beats dosage. Biochemical oscillator synchrony allows cells individually and collectively to align functions in space and time. From the firing of neurons to the contraction of muscles to the cycling of genes, life is shaped by rhythmic communication. Cells use chemical, electrical, and mechanical signals to stay in sync, echoing the beat of evolution itself. Far from chaos, biological timekeeping is dynamic, robust, and exquisitely orchestrated. Studying it offers a window into the deep logic of living systems. In embryonic development, segmentation clocks synchronize across thousands of cells to form body plans. It's like building a cathedral with no blueprint, just a shared rhythm.